Hello, I would like to thank the organizers for the opportunity to present during this year's conference, where today I'll be talking about molecular profiling in cancers from patients of sub-Saharan African ancestry. My name is John Carpten. I serve as a professor and chair for the Department of Translational Genomics at the University of Southern California, Keck School of Medicine in Los Angeles, California. Here are my disclosures. So as we consider molecular profiling in diverse populations, such as individuals of sub-Saharan African ancestry, we tend to bring this to bear under the context of health disparities, or those cancers that disproportionately affect underrepresented minorities, including, for instance, sub-Saharan individuals of sub-Saharan African ancestry, or those that are otherwise considered Black, versus individuals who might have European ancestry or be primarily of largely European descent, or those individuals that we might consider white. Looking at data from the American Cancer Society, where statistics are provided on, for instance, incidence and/or in this case, death rates of various cancers in men and women. As we look at those rates among Blacks versus whites, we can see consistently for male and female that there are quite significantly higher death rates among Blacks, particularly as we consider men, but we can also see these differences we consider women. ACS also puts out statistics on incidence rate ratio differences among Black versus white individuals across a number of tumor types. And here looking at incidence rates for a number of cancers that have significantly higher rate ratios, meaning that this is the significance of the disparity in terms of incidence rates among Blacks compared to whites. We can see a number of tumor types, Kaposi sarcoma, which is an age-related malignancy, but other cancers that in many cases are rare forms of cancer, like myeloma, gastric cancer, liver and cholangiocarcinoma. In men, we see prostate cancer, where there's 1.7, nearly twofold greater incidence rates. And also looking at, as we consider death rates, again, stomach, gastric cancer, multiple myeloma, we see cervix and uterine, cholangio, and then breast in women. In men, we see stomach and prostate, throat cancer, myeloma, cholangiocarcinoma, and then colorectal cancer. And so you can see a number of tumor types that have quite significant increased rate ratios when we consider both incidence and death rates among Blacks versus whites. Now, it's important to state that we know that these differences that we see in incidence and death rates are likely due to a combination of social and societal factors, as well as biological and/or genetic factors, as we consider the social and societal, or the social construct of race. We consider or think about the social determinants of health, socioeconomics, access to limitations and access to care, financial toxicities, which we know drive disparities, these differences that we see in incidence, prevalence, and outcomes of various forms of cancer. But we also know that there are a number of studies that are highlighting the importance of genetic ancestry and these genetic and ancestral differences. Particularly as we consider the molecular features of tumors that are derived from individuals that have various genetic ancestral proportions, either higher African ancestral proportion versus European ancestral proportion. And we know that these two things actually interact, and there's an interesting interplay between race and the social construct and genetic ancestry, and how both of these concepts are brought to bear as we consider cancer and the host and tumor biology. But we know that there's been a lot of work that has, over the last 5 decades, 4 or 5 decades, that have shown the importance and the relationship between the social determinants of health and these disparities. But there's been a significant lack of investigation into the differences in the tumor biology across racial and ethnic groups. and it's only been over the last maybe 15 or 10 to 15 years that we've begun to see studies emerge by where we're interrogating the molecular features of tumors from individuals who are largely of sub-Saharan African ancestry versus those individuals that might be of largely European ancestry. And so exploring the role of these biological differences in tumors across various diverse populations will help to broaden our understanding of the complexity of cancer in ways that hopefully will lead to the most optimal approaches for disease management for all patients. I think there's a great example as we consider the Cancer Genome Atlas, and we know that there was a significant underrepresentation of tumors derived from diverse individuals. We consider Black individuals, Hispanic, Latino, by where if that data was analyzed, there's been publications showing that, if you want to identify mutations that, for instance, have frequencies in the realm of 10% versus or 5%, that we do a really good job when we're looking at whites or individuals of largely European descent. But when we go outside of breast cancer, we really don't have power to detect mutations in these patients from underrepresented populations, given the amount of data that's available currently in the public databases. So, and this is especially for tumor types that disproportionately affect Blacks or African Americans or individuals with higher sub-Saharan African ancestral proportions: prostate, triple negative breast, colorectal, endometrial, leukemia, multiple myeloma. And so this remains an important unmet medical need in helping to determine if these genetic or genomic differences are associated with the differences that we see in incidence and outcomes displayed among underrepresented patients. So I hope to walk through a few examples. Again, this is just sort of an expansion of this plot showing, again, outside of breast cancer, we really lack the power to detect mutations, for instance, in the 10% frequency range. When we go beyond whites and we want to go down to 5%, we just completely lack power. One study actually that I think highlights, as we look across the broad spectrum of cancers, a group actually took all of the data from the TCGA, even understanding that there were limitations in the number of patients from underrepresented populations that provided tumors that were used in that study. A group led out of University of Pennsylvania, Tim Rebick and others at Dana-Farber actually deduced genetic ancestry from all patients across the TCGA, and then looked at mutation frequencies in the tumors as a function of genetic ancestry, as well as self-identified race, and these results revealed that looking at certain tumor types that included breast, head and neck, endometrial cancers derived from African Americans compared to European Americans, the tumors from African Americans exhibited higher levels of chromosomal instability, but lower levels of chromosomal instability were seen, for instance, in kidney cancers derived from African Americans. Also, they saw higher rates of P53 mutation and cyclin E amplifications in these tumors derived from African Americans. And it has been shown by others, they observed lower frequencies in genomic alterations within the PI3 kinase AKT pathway in tumors derived from Black or African Americans or individuals with higher sub-Saharan West African ancestral proportions. So these were some of the data that are supporting this whole concept of why we should be performing molecular profiling, and the justification is understanding the biology, but also as we continue to move down these precision approaches for cancer care, identifying the processes that are driving tumors across various patient populations, perhaps identifying those key trunk events and pathways, and perhaps we might learn more about the initiation and progression of cancer, again, to better manage disease from individuals across multiple populations. So I want to walk through a series of vignettes of studies that are providing this evidence across a number of tumor types, where we're seeing differences in incidence and or outcomes but also beginning to identify differences in the molecular profiles of these cancers. So as I mentioned earlier, prostate cancer historically has shown higher incidence and death rates among Blacks compared to whites and individuals from other ethnicities. Both genome-wide association studies have revealed that a number of the SNPs that are highly associated with prostate cancer risk that map to chromosome AQ24 are also highly enriched among African populations or individuals of sub-Saharan African ancestry, whether we're considering individuals from Africa, from the continent of Africa, and individuals across the other components of the diaspora. So we think about Afro-Caribbeans and particularly here in the United States. The TCGA I mentioned before was able to perform a study of roughly 330 or so primary prostate cancers, where they revealed the molecular taxonomy of primary prostate cancer, where they identified molecular subtypes of tumors that were enriched for specific somatic alterations or somatic mutations. However, within that cohort of 330 or so primary cancers, only about 25 were from African-American men. In a study of Levi Garraway and Ike Powell, published a study in Cancer Discovery back in 2017, where they performed whole exome sequencing on about 100 or so African-American primary prostate cancers and identified mutations in ERF. Actually, this gene and these mutations were not highlighted in the TCGA study, likely because of the lack of African-American or tumors derived from African-Americans in that study. And they went on to show the functional consequences of loss of ERF and its role in activating the ETS transcription factors have been validated by others in very high impact journals. Again, beginning to highlight and being among those early studies showing that there are molecular differences if we look at tumors derived from individuals from that, again, have higher proportions of West African or Sub-Saharan African ancestry. I think gene expression profiling is also of high consideration, as currently there are a number of commercial tests these are commercial clinical tests that are available to provide pre-treatment information to help determine risk of aggressive disease for cancer-related mortality in prostate cancer. However, many of the gene expression profiles that were used to determine or to create these tests were developed from largely European cohorts. And that's important because a number of gene expression studies performed in tumors derived from Blacks and or whites, where gene expression profiles were compared, have revealed that there are differences in the transcriptional profiles and in consistently seeing an increase in pathways in genes differentially expressed that are enriched in pathways associated with inflammation and inflammatory responses, suggesting that perhaps tumors derived from African Americans have a tumor microenvironment that's more inflamed, or there is a tumor signature that is driving inflammation in the prostate. And this might be somewhat different from what we see tumors derived from whites. So we need to seriously consider the development of clinical tests and not necessarily rely so much on generalization of these results, which could lead to lower precision in the accuracy of these tests in a clinical setting. So again, we need to question this whole concept of generalizability, where data that's generated from largely Europeans is used to develop tests that are then distributed and utilized across individuals from more diverse populations. And we need to perform studies to better validate across more diverse populations. And again, so we need to assess these large cohorts of African Americans. And our hope is that these studies are moving forward quickly to assess this important question in prostate cancer diagnosis and prognosis. Also, Steve Paterno, Jen Friedman, and others have published a series of studies along with Norm Lee and others where they've identified significant differences in RNA splicing in both tumor and normal prostate cells derived from Black versus white individuals, and show that when you look at differential splicing, there are as many or more differences as compared to just standard differences in gene expression. And they have published recent papers showing that specific gene spliceoforms, particularly PIK3CD, where they see a very specific spliceoform that's enriched among tumors derived from African Americans. And they're moving forward with a number of precision medicine studies, both for diagnosis, prognosis, and even potential treatment approaches based on these data. So yet further evidence that there are differences in the molecular features of prostate cancers derived from individuals of African ancestry. I happen to have the pleasure of being involved in the RESPOND study, which is being led by Chris Heyman out of USC here at the Norris Comprehensive Cancer Center, and others, where we are working with state registries to accrue roughly 10,000 African American men with incident prostate cancer to perform a series of epidemiological population-based studies, behavioral studies, as well as molecular studies interrogating the differences in the tumor and the tumor microenvironment in aggressive prostate cancers derived from Black versus white men. So we're really, really excited about this, and we're really beginning to ramp the study up. You can see all of our partnering institutions and states where we're utilizing registries to identify these men, and we're beginning to gather samples and tumor specimens, and some of the molecular profiling has actually been completed. So we're really excited about the results of this study and helping to understand the molecular features in tumors derived from Black men, particularly the aggressive cancers that we see frequently in these high-risk men. Another examples in multiple myeloma are, as I mentioned earlier, there's a significant increased rate ratio in myeloma incidence rates among Blacks, as well as death rates. And so myeloma has historically represented one of the most significant disparities, cancer disparities, both from the standpoint of incidence as well as poor overall outcomes. I had the privilege to help lead the COMPASS study that was funded by the Multiple Myeloma Research Foundation, which was a longitudinal study to accrue 1,000 newly diagnosed treatment-naive multiple myeloma patients for full molecular profiling by whole exome sequencing, RNA sequencing, and some whole genome sequencing, and very, very detailed phenotyping. And the importance, another important component of this study was that it was, or design component was that this is a longitudinal study. So patients were followed over time. So as patients may, treatments may fail patients and cancers may become more refractory, additional tumor or refractory tumors are also collected from these patients and profiled so that we can really understand the natural history of myeloma. Another, I guess, design benefit, if not whether it was serendipitous or not, was the fact that we had a significant amount of diversity in this population by where we published a paper assessing the molecular profiles of over 700 myeloma cases by whole exome and RNA-seq. And importantly, among the 700 cases, about 500 or so, 550 or so were European descent, and about 125 or 30 were actually self-identified as African American or had higher sub-Saharan African ancestral proportion, which allowed us to have a very well-powered study to look at differences in the molecular profiles across these tumors. And we revealed for the first time the molecular taxonomy and mutational profiles of multiple myeloma in African Americans using very, very deep molecular profiling approaches by where we identified these mutation frequencies and common and less common, commonly mutated genes but more importantly, we were able to compare the frequencies of these mutations in tumors derived from white versus Black patients. And we, for the first time, identified a series of genes that had higher mutation frequencies in Blacks versus whites. But what was really exciting were some results where we identified significantly higher mutation rates in P53 in tumors derived from whites compared to Blacks. And this is very, very important. And we also looked at percent European ancestry and showed that we looked at wild type P53, we saw, a distribution, but when we look based on European ancestral proportions, but when we looked at mutation frequencies, almost all of the patients that contained P53 mutation had high European ancestral proportion. 2 individuals that self-identified as African American harbored P53 mutations. Interestingly, they had relatively higher European ancestral proportions as African Americans, as the average typically hovers around 15 to 20% if you look across the United States. This is really important because P53 mutation and loss is associated with poor outcome. So these data might suggest that African American patients may have tumors with molecular features associated with more favorable outcomes. Now, this is, sort of inversely proportional to what one would think in terms of we consider the historic poor outcomes that we've seen among African Americans. But these data may suggest that equal treatment could lead to equal or better outcomes among African Americans, especially in the new era of the utilization of the blockbuster drugs, the immunomodulatory drugs, and the proteasome inhibitors that are actively being used for the treatment of myeloma. And we were excited about the results of a study looking at real world data and looking at outcomes in multiple myeloma, Rick Pazder and others, where they observed that imited proteasome class drugs may be working better in minorities, where they're seeing overall 5-year survival rates increased greater among African Americans compared to their white peers and white counterparts. And again, this might suggest that African Americans have tumors with more favorable outcome. So if they receive the appropriate treatment, they'll do as well and maybe even better again than their white counterparts. So the historical disparity in outcomes that we've seen among African Americans for myeloma may be pure socioeconomics and access to the appropriate therapies and not biologically driven. However, this does not explain the differences that we see in incidence rates, which are still quite significantly higher among individuals of higher African ancestral proportions versus individuals of European descent. And a number of studies are underway looking at differences in immunological features and immunological development, as well as genetic genome-wide association studies to look for candidate genetic variants that might be associated with the increased risk that we see in myeloma. And so we're really excited about the results of these data, and they have huge translational implications. Breast cancer, we know historically that although white women have higher incidence rates compared to Black women tend to have higher death rates compared to whites and women from other ethnicities. And we definitely consider the basal-like breast cancers or the triple negative form of breast cancer as being at least partially associated with this increase in death rate, these more aggressive cancers that are much more frequent in Black women compared to European women. Individuals have performed a series of studies, including looking at mutation rates in tumors derived from Black versus white patients. In this study, looking at breast cancers as a whole, not just triple negative, but breast cancers as a whole, and identifying mutations that had higher frequencies in Blacks versus whites, again predominated by P53. Interestingly, one of the key molecular features of the triple negative subtype, or ER negative cancers, ER negative breast, hormone receptor negative breast cancers are P53. So P53 mutations tend to be enriched in ER negative or hormone receptor negative breast cancers. They also looked at gene expression profiles and found a significant increase in CRYBB2 in tumors derived from Black versus white. And these differences persisted across many of the subtypes, including normal breast tissues. Fumi Olopati and others also went on to show, and this was one of the studies led by Fumi, interestingly in that same paper, looking at sort of the genetic contributions or inherited contributions to these differences that we see in breast cancer among Black versus white white patients. Interestingly, looking at polygenic risk scores, showing that you definitely had a higher sort of genetic contribution in the ER negative or the hormone receptor negative breast cancers versus the hormone receptor positive cancers, again suggesting that some of these differences might be likely associated with inherited susceptibility to these various forms of breast cancer, and perhaps even the tumor initiation and progression and worse outcomes that we see. Lisa Newman and Melissa Davis at Weill Cornell are spearheading, really exciting studies with a focus on triple negative breast cancers derived from Black versus white women, and doing a lot of work in assessing genetic ancestral proportions in these women and looking at molecular features of these triple negative breast cancers in women that have higher African versus European genetic ancestry, and identifying pathways that are enriched in gene expression data from women with higher African ancestry versus European ancestry. And so they're really pioneering a lot of this work and looking at immunological features in these cancers, again, clustering patients based on high African ancestry versus low African ancestry, and identifying various immune effector cell subtypes that might be more enriched in these triple negative breast cancers. So we're really looking forward to the continued work and results of Lisa and Melissa. They really are doing an amazing job of collaborating and helping to build infrastructure in Africa for breast cancer research. Our group has been focusing on nuclear alpha-catenin and its role in DNA repair, showing that homozygous deletions at the alpha-catenin locus are enriched in the triple negative breast cancer subtype and are more enriched among tumors from Black women. We also show that loss or low alpha-catenin is associated with a significant reduction in survival probability, and tumor cells that don't express alpha-catenin based on CRISPR-Cas9 knockout are far less sensitive to many of the DNA damaging agents associated with or used in the treatment of triple negative breast cancer, meaning that these tumors, these triple negative breast cancers that might be, that have loss of alpha-catenin and we believe are enriched among tumors derived from Black women, are far less likely to be responsive to chemotherapy. And then finally, Fumi Olopati and her group profiled almost 200 breast cancers from women from Nigeria, and interestingly showed that tumors from Nigeria that were hormone receptor positive were characterized by increased homologous recombination deficiency, pervasive P53 mutation, and greater structural variation, which are features essentially more associated with triple negative or basal-like breast cancer. So really amazing and interesting results in characterizing these tumors derived from women from West Africa. And even in colorectal cancer, where we know there are differences, our group in collaboration with Xavier Lohr and Nathan Ellis performed a whole exome sequencing and methylation analysis of colorectal cancers derived from African Americans, and were able to provide the mutation frequencies as 45 African American microsatellite stable colorectal cancers from African Americans, and identify differences in mutations. And what we saw was an interesting a series of cases that lacked APC mutation, where we see APC mutations were higher in the TCGA versus the cancers in our study. Interestingly, these APC, these tumors that lack APC mutation were also associated with a younger age at onset. And we also have identified unique epigenetic signatures among these tumors that lack APC mutation that we see among African Americans. And this work has been followed up by a funded R01 grant that's being led by Nathan and his colleagues. I think as we continue to consider this work, it'll be important for us to continue to work on the development of collaborations and international consortiums across the diaspora, Africa, the Caribbean, the US, South America as well. I think another consideration is clinical research on the continent of Africa, building infrastructure for clinical trials and patient engagement, clinical pathology through, again, consortiums and collaborations, biobanking, research pathology, tissue repositories, and database management need to be bolstered both in the States as well as across the African diaspora to help build up the collection of samples that are needed for this detailed molecular profiling. And data science and clinical informatics, as well as biomedical informatics, where we believe there's a real opportunity to collaborate with our partners by where this would require the access to computing infrastructure and not more expensive and complex molecular profiling technologies. Finally, the technologies for genomic and genetic and genomic profiling, as we consider DNA, RNA, epigenetics, and even some of the more advanced technologies for single cell and spatial transcriptomics. And again, building this infrastructure through collaborative grants, workforce training, and fellowships. So in summary, health disparities are real. There are differences as we consider incidence and outcomes among Black versus white patients. We know that these differences that in tumor biology have not historically been well understood. However, there's mounting evidence from recent genomic studies, many of which I've shown today, demonstrating that these differences do exist. Profiles are not likely going to always be generalizable. So we have to diversify these cohorts, diversify these data sets so that we can better understand the disease etiology across these different populations and how these data could provide unique opportunities for improving disease management, potentially leading to decreases among certain known and validated cancer health disparities. And we must also ensure patient diversity in discovery and validation research and in clinical translational research to increase our ability to move to be more thoughtful and impactful towards developing therapeutic approaches that are more personalized and have a broader applicability across individuals. We're really excited about the new integrated approaches, such as those that we're taking with our response study to integrate across behavioral science, environmental science, understanding social stressors and social conditions in the built environment, and how this impacts on increased stress can impact detrimental physiological conditions, epigenetic changes, transcriptional changes in protein modifications and lipid metabolism and other things, including chronic inflammation, that then drive these pathways, these cellular pathways that we know are enriched among those that are considered the hallmarks of cancer. And so a number of studies, multidisciplinary, interdisciplinary studies are being conceived and designed, and some are underway to help us better understand this relationship between, again, the social structures, the social determinants of health, and how they drive some of these molecular differences that might be influencing differences in incidence and outcomes that we see among Black versus white patients. So we're really excited about the prospects of these new study designs. So thank you, and I'll stop there, and I'm really looking forward to the Q&A section tied to this scientific session for the conference. Thank you.